Hey, so anybody got any questions? I'm assuming you can hear me. Yeah? Anybody? I didn't check my mic. All right, so the question coming in on the chat is absolute refractory period versus relative refractory period. I'm assuming you can hear me? Yes? Somebody type yes in chat or jump on the
I can hear you.
microphone and say yes.
I can hear you.
Professor Hoffman, my question was mainly like which gates are opened and closed during those two sections, during absolute refractory and relative refractory. I feel like in the video you said don't worry too much about it, but I just got a little bit confused. I feel like at one point you said absolute refractory, no, all the gates were closed, which is why there couldn't be any type of like incoming stimulus. And then in relative, maybe one part of the double gated channel was open, but the other part wasn't. I don't know. I just need some
Yeah,
clarification.
so I'm going to do this. So you should be able to see the screen now with the double gated sodium channels. And if you look here, you'll notice there's two gates. And for the two gates, one of the gates, right, is down here at the bottom. So if you look, I'll just kind of, not circle it very well, circle it, and the other gate's right above it right there. So depending on the configuration of the gates, it gives us the absolute and relative refractory period. So it doesn't matter to me that you know that this one's open, this one's closed, or both of them are open, or both of them are closed at a given point in time. Obviously, to have the action potential, you have to move sodium from one side of the membrane to the other, so they both have to be open. But if you look at the next slide, I can find my cursor. So if you so if you look at the next slide, this kind of shows the difference. And again, we're going to consider the absolute refractory period, the time where you go from the peak to down to about minus 30 millivolts, so right here. And not where they show it on the blue, it actually lasts shorter than what they show on this one. And that's when the channels are a certain configuration where they're basically locked. And think of it like a time vault on a bank that doesn't open until, you know, Monday morning at 8 o'clock or whatever. So those won't open no matter what. So that's what we call the absolute refractory period. And then at about minus 30, all the way back until it gets back to the resting. So all of this right here, that's the relative refractory period, which says that we need a bigger than normal stimulus to do it. And again, it's just the difference. The channel configuration changes, but you don't have to know how it changes. Does that make sense?
Yeah, thank you.
You can say no. Does it?
No, I said yes. Could you, I don't know if you could hear me.
No, I heard you. I heard
Oh.
you. It sounded like it was a very reluctant yes. Like, I don't want to ask for a follow-up. You can ask for a follow-up if you want.
Oh, no, it's okay, really. I
Right.
think I get it. I think I was just hung up on feeling like I needed more to know more detail than
you do. No,
I do.
just be able to identify the absolute refractory period, which is associated with the repolarization phase, the first part of it, and the relative refractory period, which is associated with both the repolarization phase and the hyperpolarization. Exactly.
Okay. Yeah. Okay. I'm good.
Okay.
I have a question about the phases, actually.
Yeah.
So some of the slides say that there's like 5 phases. Some of the slides talk about 9 phases, and they're often called different things each phase. So I'm kind of wondering, like, how do we consolidate that? Like, what do we need to know? I mean, I understand clearly like the depolarization, repolarization threshold and stuff like that, but
So that's good.
okay.
Let's kind of this one's oh, of course it would help if I actually shared it. So I'm going to share, this one is probably a good one to kind of know. So looking at the kind of the phases of the action potential, the places that we care about, and then, you know, this has those, I think it's probably the 9 you were referring to. What we want to look at is we want to look at from number 1 to 2, right here. So I'm just going to make it thicker so you can see it. Just make it red as well. So, no, I don't want it red. It's already red. I'll make it blue. So right here, in this area right here from 1 to 2, that's the resting membrane potential. Okay, what you should understand about that is it's minus 70 millivolts. That's a number you should know. It's determined by the relative permeabilities of sodium and potassium primarily. The cell is mostly permeable to potassium right now. Most of the channels, relatively speaking, for sodium and potassium, like 97% of the channels are open for potassium, and only 3% of the channels are so open for sodium. So there's a disproportionate amount of potassium channels that are open during rest. Okay, I'm going to change the color. We'll go to this kind of ugly orange thing. And then from 2 to 3, if I can do it, from 2 to 3, that's our graded potential. Okay, graded potentials can be excitatory or inhibitory, right? And the graded potential is what actually causes the action potential. To get an action potential, we have to get a graded potential, an excitatory one. Those are called EPSPs. We can talk about more of those later if you want in terms of detail. That has to reach threshold. Once we reach threshold, we open a double-gated sodium channel, and it's a voltage regulated channel. So we've talked about channels before, and these double-gated channels only open when they reach minus 55 millivolts. And so we have to reach that. That's what we call that threshold. That's a good number to know. And once we do that, then we get the, which was kind of talked about a second ago, depolarization. So I'm going to draw on green as best I can up to number 5. That's our depolarization phase, right? Oftentimes people call that the action potential. The action potential is actually the whole thing, but that's our depolarization. And the reason why we call it depolarization is we start at minus 70 and we go towards 0. So we're going from a less polarized state towards 0. So that's what we call depolarization. That occurs mostly because we are opening a large amount of sodium channels, but we're also opening a few more potassium, not a few, actually more potassium channels. But because there's more sodium coming in than potassium leaving, think about your checking account. If you had more money coming in than you were spending, it would get more positive. And so that's what happens here. The peak of the action potential, if you look at number 5, right, is right at minus 30, sorry, plus 30 millivolts. So this is number 5. So that plus 30 right here for the peak, that's what you should know is the peak of the action potential. So that's another number you should have an idea. Once we reach the peak of the action potential, we start to close sodium channels and open a more proportionate number of potassium channels, which means now we have more potassium leaving, so more positive ions exiting the cell than coming into the cell. And so now the depolarization, right, now goes negative. So now, because it's negative, we call it repolarization. So that's that level right there. So that's repolarization from about plus 30 to minus 30. And that's when we have the absolute refractory period. And again, that's because the channels have now, those double-gated channels have gotten into a specific position where they won't open no matter what. And then continuing down the repolarization, which means we're just going back to a polarized state, is we're just going to keep on going down through hyperpolarization, which I'll talk about in a second, back to rest. So that lilac purplish color, whatever you want to call it, that is the relative refractory period. So the pink is the absolute refractory period. That means we can't open a channel and get another action potential no matter what. And the purple would be the relative refractory period, where we need a bigger than normal stimulus to do that. So the absolute refractory period happens during the pink, so it's only associated with the repolarization. And the relative is the purple, so it's repolarization. And then once we get to number 7 down here, right, we're going below rest, so this is called hyperpolarization, or sometimes called after hyperpolarization. So this is due, this hyperpolarization is due because there's more potassium channels open at that time relative than when it's at rest. And again, just to kind of review that idea, when I discussed on one of the videos, actually did a couple times in a couple different ways, we had 2 magic numbers, the plus 30 at the top and the minus 90 at the bottom. And those are what's called the equilibrium potential for sodium and potassium. And that means if the cell was 100% permeable to sodium or potassium, so let's take potassium first, that's the minus 90 on the bottom down here, minus 90. If we're 100% permeable to potassium, that means that all of the channels that were in the membrane were open for potassium, which means potassium would leave. And as potassium starts to leave, the inside of the cell becomes more positive. And because, the inside of the cell becomes more negative for potassium. So the inside of the cell becomes more negative. That's why it's minus 90. So as the inside of the cell becomes more negative as potassium leaves, it starts to be attracted to the positive charge for potassium because it's a cation. And so it gets harder and harder and harder for potassium to leave until eventually the charge inside the cell is equal and opposite to the charge, the chemical gradient of potassium leaving. And so that's called the equilibrium potential. And for potassium, it's at minus 90. For sodium, since it's high concentration inside the cell, low concentration outside the cell, it's actually not here. But this would be, trying to write it in neatly, make a better plus sign, positive 60 millivolts. So positive 60 is where the equilibrium potential for sodium is. So if we had only channels open for sodium in the cell membrane, sodium would come in. As it came in, because it's a cation, it makes the cell more positive. As it makes the cell more positive, it starts to push back against the positive sodium coming in. So that's called electrostatic repulsion. And the electrostatic repulsion eventually gets big enough when you get enough positive ions in there that it tells sodium you can't come in anymore because we're too positive. And that occurs at positive 60 millivolts. So that's called the equilibrium potential for sodium. So that kind of constrains our graph a little bit, but that's sort of what you should be able to tell me. So the magic numbers that you should know, minus 90, the equilibrium potential for potassium, plus 60, the equilibrium potential for sodium, minus 70, the resting membrane potential, minus 55, the threshold voltage, plus 30, the peak of the action potential, about minus 80 for the after hyperpolarization for the action potential. And that's kind of what you should pretty much understand from that. So I'll clear all that. And again, there's going to be I'll post this up on Canvas or link to Canvas on YouTube. And the one up from last night's up there too. Does that make sense? That was more than what the question was asked, but I think a lot of that helped explain everything.
Yeah, thank you.
All right. Anybody else? What else? What else we got? You can type them in if you don't have a microphone. You can. All right. To clarify the first essay question. So the question is from the question that we have of the take home. And this is kind of actually a little confusing. Matter of fact, I anticipated that this would come up. So let me do this. So I've got this thing ready today because it came up yesterday, and hopefully I can share this with you. So there it is. So this is the answer from the group problem, which doesn't really exactly answer the question that we just had, but sort of does. So the question was, you know, when we get 38 ATP total, where do we get that from? So we'll come back to this schematic in a second, but where it really comes from is this right here. So let me kind of show you some stuff on that. So when it's asking for the 38 total ATP, if you look at this one, this is the answer to the group problem. And the group problem actually asks for 40 total ATP. So if we come here, the total we're talking about is each row, right? So glycolysis, activation, Krebs, and then we total each row. And then we have an ATP total for the total of each row. So this number right here is the number we're looking at. So for this one, it's 40. And I think, you know, those of you who've been looking at it and, you know, fairly significantly in the last week or so, probably, and you may not remember this if you haven't looked at it since we talked about it in class, but the total number of ATP we get using the malate aspartate shuttle, which is seen up here, right? So here's malate aspartate shuttle, gives us 40 total ATP. So when we look at that number, right, we'll see, oh, that's not right, because when the question for the exam says you got 38 total ATP. So to get 38 total ATP, you would have to use another shuttle. That would be the glycerol phosphate shuttle to get to 38 total ATP, right? So you wouldn't use this, but you would basically kind of have the same idea with this and make sure you had the numbers correctly. So what the question was, well, if I asked for, like, you know, 16 or 18 or 14 ATP total, I might ask you for a particular one, like how much do you get in glycolysis, or how much you get in activation, or how much you get in Krebs, but I would not ask you to put two of them together unless I ask you for all of them, which is already total at the bottom, right? So if I said in let's say glycolysis, what's your total ATP using the malate aspartate shuttle, then you can just look at this table and say, oh, that's 10, because I see it right there, right? So there's my total ATP column, and that's 10. Or activation, you know, would be 6, or Krebs would be 24. And we'll kind of give examples of kind of how to figure a few things out before. So it'll be similar to yesterday, because I know that some of you were here yesterday just looking at the names, and a number of you weren't. So we probably want to, you know, visit that again. But that's what I'm talking about for that. So let's see. Yeah, so the question is, the exam is asking for total ATP, not net ATP, right? And so the problem is when I'm sharing, I can't see what you guys wrote. So let me go back and share that screen again. And so what the difference is, right, the question asks for 40 total ATP, not the 38 net ATP, right? So yes, it makes 38 net ATP, but since it's total, you have to go to the other shuttle, right? So we don't want to use this one. We want to use where it says 38. And hopefully you remember, because of this, right, in glycolysis, we make 2 NADH no matter what, right? But in the malate aspartate shuttle, because it's a passive transport mechanism, it becomes NADH in the mitochondria. Since NADH gives us 3 ATP in the electron transport chain, right? And I have 2 of them, I get 6 indirect ATP there, right? But for the skeletal muscle, right, using the glycerol phosphate shuttle, this goes away, right? And because it's an active transport mechanism, we only have enough to make 2 FADH2s. So this becomes 4 indirect ATP, which means eventually makes our total ATP, it's hard to write with the mouse, 38. So that's what we're looking at there. And that's the big difference that you're going to have to discuss and mention. So hopefully that makes sense when we look at those things. All right. So does that make a little more sense?
Yeah, I just needed to clarify that you really were asking for total ATP,
so that's the column you're looking
not
at.
net ATP.
Yes. Yeah. And
Okay.
so to kind of follow that up, right, I'm going to be really specific on the exam, right, and make sure you understand it. So it's semantics. What's the difference between net and total ATP? Well, it's not always different, right? So as an example, let's again share that table to make this point. So on this table, if I said, okay, what's the total ATP in glycolysis using the malate aspartate shuttle? You would say, oh, that's 10, because I get 4 direct, right? So right here I get 4 direct, and then I get 6 indirect, and that adds to a total of 10. Well, what's the net ATP for glycolysis? Well, you got to take the total, the 10, and subtract the U's 2, so we get 8. But if I ask the same question for activation, I said, what's the total ATP you get for 1 glucose molecule in the activation step? Well, you come over here, and so I get 2 NADHs, right, which gives me 6 indirect ATP. I don't get any direct ATP out of that, so I don't have any ATP directly made. So that gives me 6. So what's your net? Well, we didn't use any for activation, so the net is still 6. So in this case, they're the same number. In the case of glycolysis, they're different, and that's because we have to use 2 ATP to start the whole thing. So the point that Jessica's making about the semantics, right? Make sure you understand the semantics and you're reading the question carefully, right? Is it direct or indirect, or is it total, or is it net? Is it for 1 turn of the Krebs cycle versus a whole glucose molecule? All of those different aspects are going to make it a different answer potentially. And so, you don't want to, answer it one way where, I have 2 questions. Here's like literally, there's going to be like 4 or 5 questions on the exam that's going to ask you exactly for these things. How many ATP do you get in this process? How many do you get indirectly? How many do you get directly? How many do you get net? How many get total? And so you have to make sure you understand exactly what I'm asking for in terms of that process. Does that make sense?
Yeah.
Okay. Anybody else? All right. So while you're thinking of other questions, or if you want more expansion on any of those concepts, I had a question in the chat. And that's interesting. That's the same question they asked yesterday about tonic activity. We'll come back to that in a second. But one of the other questions was, why does our fever? Why does our when we have a fever, why does our set point change? It's really the question, right, is really answered best the other way around. Our set point changes, and that's why we actually have a fever. The fever is actually a reaction immunologically in our body, right, to try to fight off whatever infection we're having. So as an example, one of the hallmark symptoms of being infected with the coronavirus right now is a high fever, right? As a matter of fact, we still have, despite what seems to be what the government officials are telling us, there still seems to be a large number of hospitals and clinics that do not have sufficient kits in order to test for the virus. So one of the things they've been doing is they've been saying, okay, we're not going to test you unless you have a high fever. And it's been different for different ones, but usually, they're expecting it to at least be 103 degrees or higher. So the reason why that happens is when you get infected, your body actually takes that set point and shifts it up to a higher point, where you regulate your temperature now. So instead of regulating it at 98.6, your body might regulate it now at 103 degrees Fahrenheit. And the reason why is that from an immunological standpoint, you're much better off physiologically to fight off that virus or whatever is making you sick. And so now you actually regulate your temperature at a high point. And so, oftentimes you don't feel like you're really, really hot because of that, although certainly, you're going to feel other things. So it's the set point that changes in terms of that from a physiological standpoint to make the temperature go up, because our body will better fight the what's doing this. It's partly due to also the cytokines and other things released in terms of cells from the body, or even potentially even cells for making you sick, that raise the temperature. But primarily it's us resetting the set point to a higher temperature, which gives us a better ability to fight off the virus. And so that's one of the changes we see. The question in terms of tonic activity. This is same question we had yesterday, and I'll kind of do this. So I'll share another screen with you. So basically, and hopefully you've seen them. I have changed the group problems from what's originally been posted, right? And some of them, 4 of them are the same because we've done it or talked about it. And 2 of them were things that I took out because of officially Monday was not an instructional day, and Tuesday earlier this week. So since we had one less technical instruction day, I took out some of the information that we would have done had we been in class. To be honest, it was 2 of the harder questions on this. So I thought we'll give you a little bit of a break here. And I also thought it made sense to try to finish and wrap up what we were talking about in class, and then just add a few things that I wanted you to do. So there are new notes posted. There are new PowerPoints posted. And I put them at the modules right at the beginning of the Canvas section when you go into it, so you can't miss it. But those are the things that you want to probably look at. But don't forget, there's still stuff on the original module that would help. And I think I removed everything that wouldn't. But as an example, there's like a practice exam on it that will help for some of it, especially for the receptor things. I think that it's important for many of you going into a medical field to understand the neurotransmitters and receptors of the peripheral nervous system. And that's the only time we have an opportunity to talk about them. So I did hold you responsible for those, and they're in the end of the thing. But long story to get to this screen. So the question was asking about the tonic activity. And so there are 4 postulates that Cannon came up with in order to describe homeostasis way back in the late 1800s and early 1900s. These have no particular order, although I number them 1 to 4. It's not like the 4th one you couldn't say first or whatever. But here's the 4 postulates. We'll start with number 2 because that was what the crux of the question is, and might as well kind of just finish the idea, especially since it isn't a group problem. And so the first one we're going to talk about is there's always a level of tonic activity. There are 2 main methods to control homeostasis. We mentioned that a little bit, the nervous system and the endocrine system. And when we have nervous integration to places, and in terms of the endocrine system, especially in terms of hormones, there's always what we call some level of tonic activity. It means there's always some level of action potentials going to a blood vessel or to another neuron, like the Renshaw cells, or to a gland to release sweat or whatever it is. So there's always some activity. It's never completely off. And the reason why you have this tonic activity, we believe, is there may be instances where you're already sweating very little, but you want to sweat even less. And if it was off, you couldn't have any more than off. But if you're sending only, 1 ax potential every 3 seconds, now you can tone that down by sending 1 ax potential every 4 seconds. And that would decrease the level of activity. So it always gives us some level of control. So it's almost like, think of it as a radio that's always on, but you can just turn the volume up or down, but you can never completely turn it off. And that's not a kind of a bad analogy. In terms of hormones, we all have hormones in our body, and we all have the same hormones, but we just have different levels. As an example, one of the things I was reading again with COVID-19 is you see almost a 2/3 to 1/3 mortality rate for men compared to women in terms of this disease. And we actually saw this back in 1918 with what they call the Spanish flu. It was really an H1N1, and it was erroneously called the Spanish flu because Spain was neutral during World War I, and they actually reported deaths from that. Most countries didn't because they were afraid if they seemed weak, they would get invaded. So it didn't start in Spain. It more than likely started in Kansas at a military base. And there's probably a virus that was passed from pigs to humans. And then the military base, when the soldiers moved out and went and traveled throughout the world for deployment, then they brought that everywhere. But we saw a lot more men die than women. And for SARS and MERS, you saw the same thing, a higher proportion of men than women. And so the point about that was that we all have levels of estrogen. It's believed that estrogen might be part of that protective factor, but women obviously have more estrogen than men. And so it certainly means that, men typically don't have a whole bunch of estrogen, and typically it doesn't have much of a physiological effect, but there's always that tonic control. On reverse side, as an example, I guess the same idea. I mentioned in class about HCG. So HCG is the way we test for pregnancy, like in a home pregnancy test, like an EPT or something. And that measures the level of HCG. So women who are pregnant are going to have high levels of HCG, but there are some women that would test as a false positive because their HCG levels are high enough that stimulate the test and get a positive, but they're not pregnant. And in fact, some men might have a higher level of HCG, although typically it's not high enough to have a physiological effect. It's always there. So that's what tonic means. It's always there. It's just how high or low is it? What's its firing rate? That determines basically what the physiological response is. So that's one of the postulates. Going back up, the autonomic nervous system plays an important role, right? So we know that most of the things are mediated below our level of consciousness. It's not like you drink a soda and then go, hold on a second, I have to, release insulin to start taking my blood sugar away, right? It actually does that automatically. So the ANS plays a tremendous role in that because it works below the level of consciousness. And the autonomic nervous system, for example, controlling your blood pressure, adjusts heart rate, stroke volume, and total peripheral resistance. We'll see that in the next section for that. So that was one. The third one we'll talk about is there are antagonistic controls. We've seen that from literally the first week of school, right? So again, talking about whether it's blood glucose, where we have insulin that makes blood glucose go down, or glucagon that makes blood glucose go up, right? We can move it in either direction from the set point from that. And then the last one is it's the receptor that gives the effect, not the chemical. And so, we have, levels of hormones flowing around our body. But as an example, when you get scared, epinephrine, or, you can also call it adrenaline, it's the same thing, will not affect your skeletal muscles at all. Why? Because there's no receptors there. But what it'll do to your blood vessels in your heart, it'll actually cause them to dilate. So in your cardiac muscle, at least the blood flow through it, it causes dilation of it. It also increases the strength of contraction. It also increases the heart rate, and also increases how fast the action potential moves through the heart. So all these things happen, but the blood vessels in the heart dilate, and that's because they have what's called a beta-2 receptor. And that allows more blood for the heart to provide more oxygen. So when the heart starts working hard when you're exercising, you can, promote the aerobic metabolism. Your heart's not very good at anaerobic metabolism at all. But in your skeletal muscles and other portions of your body, that's actually going to cause constriction of vessels initially. And so that constriction of blood vessels is going to reduce blood flow initially, Rose. And that's because it's the exact same chemical, epinephrine, adrenaline. But in the periphery, there's what they call alpha-2 receptors. And so it shows a good example of why it's not the chemical, it's the receptor that has the effect. So the exact same chemical, epinephrine, in one blood vessel in the heart causes dilation, but in another blood vessel in the periphery, it causes constriction. And that's because they're two different receptors. So it's the receptor that gives the effect. So a quick summary, the 4 canon postulates, right? The ANS plays a major role. There's always tonic activity. There are antagonistic controls, and it's the receptor that gives the effect, not the chemical. So that's kind of what we're looking at for that. All right. So what else?
This is just a general question about the exam. So it's 50 multiple choice questions, and then we have a couple of shorter essays, and then the part.
I thought you said 49.
Yeah, 50, 49.
I
Yeah,
thought
close
you said
enough.
49.
Yeah. So.
Okay, this is why I was clarifying. And so the part that we're doing live tomorrow is 49 to 50 multiple choice, and then it's like a couple of
I think it's
short
3.
essays.
So there, I got 3 short answers that I thought you could type with words pretty well, where you wouldn't need to draw pictures, you wouldn't need to make tables, you wouldn't need to do diagrams, that you could just pretty much answer them in just word text format that you could type in. There's 3 of those. There were 7 points apiece.
Okay. And the other 30 points of the exam is the quote take home part?
Yes.
Okay.
And so there's 3 questions, right, that there were 7 points apiece, so that's 21. And then I have 49 multiple choice questions that were going to be multiple choice. They were going to be some of your fill-ins, and they were going to be some of your matching. And so I converted what would have been on the test, not all of them, but some of them. And one of the things that I got from feedback from students that took the lab quiz, which everybody did, or most everybody, but the information I got was, well, it took me a lot longer than I thought. And some of it I could tell were people, especially for that, like that molarity question, right? Where they were trying to format it or trying to figure out how to show their work for how to get to moles, then millimoles, then milliosmoles, and then tonicity. And I think that just messing with that format took some time for people. So that's what I was trying to prevent for this one. But I think for some of it, right, and the 2 questions that I asked on take home, I think you really probably want to draw stuff, and whether it's a schematic or a table or a picture or something, that's really hard to do online during an exam. So that's why I split it up that way. Plus you still have the normal hour and a half that you would have anyway. And I actually gave you an extra couple minutes to log in, to log out, right? So, but it will log in, as you guys saw, some of you saw on the lab quiz, it will log you out at the time, right? And I don't have any way to prevent that. It's the way Canvas is programmed. So when your time's out, your time's out, you're done. So make sure you get done. So I gave you an extra couple minutes that, hey, when you get close to, 9:30, if you're in the morning class, or 7 in the night class, it's just going to disappear soon. So start working hard. So that if you take each of the multiple choice or the matching or what used to be the fill-in, and I made questions out of it. So I got 49 that'll appear on the exam, and then the 3 7-point essays, that would be 21, that would give you 70 points. and then that take-home portion is worth 30 points, right? Also, hopefully you've already seen, I posted an assignment this morning, 5 points. A couple of you have already done it, where I just want you to learn how to submit a PDF file on Canvas. And I think it'd be so much easier for all of us if we just got the PDF file submissions on Canvas. And I think that what you're going to do for the take home is you're going to probably draw something, because I asked you to do it by hand. You can type in your answers if you want, your text, but if you're going to draw a table, or you can do that online. That's not too hard. But if you're going to draw the schematic or something, you really should draw it by hand, so that I can see it, and I want you to kind of go through it and show me you did it yourself, basically. That you're going to want to have some sort of way to take a picture of it, get it into PDF form so that you could submit it. And so I put a little assignment up that's due on Tuesday night, just to drop something to me through that link, because some people have never used it before. And a couple of people did already and said it was pretty easy, and they never done it before. So it's just another thing we have to figure out. But I'd rather have you figure it out and try to get caught up, so you're not worried about it. So okay, I don't have to worry about my submission. I know how to do it if you've never done it before, for a little 5-point Canvas thing, than worried about 30 class points on an exam. So together, the 70 points and the 30 points would be 100 points total, and that would give you the number of points for 100. Does that make sense?
Yeah, thank you.
So with the PDF, we're just uploading anything.
Yeah. So you probably want to think about, before you do it, what you're going to do answering those 2 questions that are worth 30 points, right? So, it'd be really easy to type a little message on Word, save it as a PDF, and then attach it, right? And if you're not going to do that, you might want to do that. But if you're not going to do only that, the idea is to do what you're going to do maybe on Wednesday. So maybe take some sort of picture and figure out how to get it into PDF form and submit it. So it shouldn't take that long, but I don't care what you submit. I got, a couple messages about life as being quarantined. And I got somebody else that, took a picture of, hey, this is my backyard. It looks pretty cool right now because it's kind of spring. So whatever. I'm looking at them, but I'm not grading them for content. I'm just, did you do it? And if you can't figure it out, then let me know, and we will go ahead and figure out why you can't do it and try to find a way to do it. So
Okay, thank you.
Make sense there?
Yes.
Yeah. All right, and you can always, email me, and we'll have some office hours, like I said, scheduled, this week, and I'll post the schedule of what you should be doing, and then I'll even have some over spring break, because most of you will probably be worried, at least starting to think about, because spring break's probably not any different for most of you now than it would be this week than next week. Although I'm not going to give you any assignments, I'm going to post some stuff up, and if you have questions, you can always do those things from home. Even if it's spring break, I'll still be here, right? We're not going anywhere. So any other general questions about the exam? It'll be very similar to what I would have given anyway. Just some of that stuff won't be on there because we didn't do it in class because of that non-instruction day we had. All right, so I'm going to move on. We always come back to anything you want, or if you want more clarification, you want more detail on something. So the question was, why the complete oxidation in the summary using the glycerol phosphate shuttle is NADH 0. So it's not exactly 0. We'll look at it here. So again, we'll come back. We'll come back to this. So we'll share the screen again. So again, this table right here, shown here, is for, again, you can see at the top, and this is again the group problems, the malate aspartate shuttle. If we move up, so here is the schematic for that, right? So in terms of the schematic for this, we see that in this case, the NADH, trying to underline it, not cross it out, the NADH that we made in glycolysis, right? When it goes and enters the mitochondria, it's NADH still, all right? So that's what happens with the malate aspartate shuttle, right? So this would be in liver cells is what we're responsible for. And the NADH that we made in glycolysis becomes NADH inside the mitochondria. So each one of these, right, would be worth 3 ATP. It is so hard to write with the mouse. So 3 ATP. And so, and looking at the column, it would be 3. But in the glycerol phosphate shuttle, right, even though we still make the NADH, right, so if we're in skeletal muscle, then it becomes, once it gets in the mitochondria, an FADH2. And so we make an FADH2. So even though we made NADH, right, when it gets into the mitochondria, it gets actively transported, so we don't have enough energy left to pass it to an NADH, so we pass it to an FADH2. So the column for glycolysis becomes 0 for that, but the actual column then would happen, in this case, the NADH would be 0, but the FADH2 would now be 2. So we still make it, but now at the end, it now becomes FADH2. And so that's what we're looking at for that. So it's technically, right, this gets crossed out, and now we've got 2 FADH2s, which means if you do the total, this is going to be 8 at the bottom, and this one, because we've got 2 more, would be 4, and then this would be 32 because we'd have 2 less ATP, and this would be 38, and then this would be 36. And if you look at that schematic that's on Canvas, right, so you go back to, you know, scroll way down to the middle part where there's the stuff for lecture exam 2, not the new stuff that I posted, that's just the changes for the because of the online stuff. You go back, you can find the schematic for 38 total ATP using the glycerol phosphate shuttle. So we just don't have that NADH anymore because we're making FADH2. So that's why it goes away, which Glenn did a good job answering anyway on here. So any follow-ups for that? All right, so what's the difference between feed forward and feedback, right? So feed forward is a physiological phenomenon where we have things happening before they actually have stimulus to cause them to change. So as an example, if you got ready to exercise, especially if you had, we're going to do a race or an event, your body will start to increase your heart rate and your respiration and things like that, so that it takes less time to reach the maximum that you're going to need in order for performance. And so that's the feed forward. Feedback is when you, and it's usually when it's inhibiting a process. So Feedback would be when you take a product, especially in metabolism, and that product will feed back and slow down the production of it. So as an example, glycolysis is controlled somewhat by what they call feedback inhibition of ATP. If we make a bunch of ATP, there's no reason to keep on converting sugar into energy from glycolysis because we have a bunch. So ATP will actually feed back on the first enzyme of glycolysis and slow it down or inhibit it. So it's to kind of use a term we used before, it'll be an antagonist and slow that production of ATP down ultimately. So feedback usually is to inhibit a process so you don't make excessive production of something. And feed forward is to accelerate a process typically to have it work better, more efficiently, faster in terms of that. So that's the difference between feed forward, feedback that we see. Just kind of looking at chat again. What time will this be posted on YouTube? As soon as I can. So whenever we're done, I will, it takes a while to render, right? So I am literally recording it right now. And when we're done, I'll stop recording. It'll take a while to render so that it's in the video format. Then I'll upload it to YouTube. That takes a while. And then in YouTube, it has to render it there. And then once I get that, I will copy the link onto Canvas as soon as I can. I got, most of the afternoon, devoted to working on this class. So as soon as it's over, I will post it on YouTube. The other one from last night's already posted. Again, it's in that section where it has the different things on Canvas. It is in the modules from last night, it is in the remote lecture file. So it says Physiology Pop-Up Shop 1, Online Office Hours 3/21/20. And I'll put this one right below it. It's the second module once you call it up. So that shouldn't be an issue. I don't know. Yeah. So here it is right here. So here's where the one from yesterday is, right? And I'll just pop up the next one right there below it as soon as it's done. So as soon as we're done, I'll work on it, but it takes a while to render. And partly I'm doing it this way because when people go directly into Zoom, then it sometimes takes up to a day to get that file back. And I am recording this directly onto my hard drive so that I don't have to do that. So I don't have to wait for the link. When somebody did it yesterday, no, I guess Friday, he said it took 17 hours to get the YouTube link on where to link it from. Not the YouTube, the Zoom link on the video. So I was just trying to shorten our time. In addition, just, there's other stuff on here. Look, hopefully you're paying attention to Canvas. I'm trying to send you more information online when I think it's important through the Canvas portal, but pay attention to Canvas and make sure you're looking for that stuff. That way you don't miss anything. So we got the also a couple other things that you can look at, and also I don't know, and you're welcome to do this, right? If you go to the group chat, right, and you don't want it to go to everyone, you can message literally anyone in the chat, right? So that nobody else can see. So you can do that as well. If you, I don't know, if you don't want people to see what you're asking, I don't know, or maybe you want to say, well, I don't think James knows what he's talking about. You don't want to tell everybody that, then that's fine. So when we look at a neurotransmitter, we have to stop a signal, right? So let's kind of look at, what are the ways we terminate the pathway. And remember, we don't want signals to continue on forever. And we want to be able to terminate a pathway so that it doesn't continue on. And so here's, again, and that's actually, in some cases, bad when we continue to have a pathway that doesn't terminate. So here's an example of how we can terminate neurotransmitter activity in a chemical synapse. So just looking at them, we have what is known as active removal, right? So that's number one, active removal. And that's when we literally pump it away from the site. So as an example, in skeletal muscle, and we talked about it in lab, we said that in order to have muscle relax, we have to pump the calcium away from the site. So that's active removal. It actually takes more energy to actively transport calcium back to where it's stored than it does to physically contract the muscle. All right. The second one is active degradation. And so that's shown. So the first one, the active removal is shown here. Sometimes it's hard to find my cursor here. There it is. So there's number one, active removal down here. Here's number two. That's the one where we degrade an enzyme. So we mentioned, just as an example, acetylcholine esterase. So if this was a synapse that released acetylcholine, so if those little red things were acetylcholine as a neurotransmitter, then this enzyme would break it apart instead of little pieces, just two, and then activate it. So that's a common way. Number three, right, pretty common, is it just diffuses away, right? So it either goes into the bloodstream there, number three, or it just goes in the interstitial fluid there. And it just, no longer has an effect because it's not by the receptor. And the other one is have something to do with the target cell receptor stuff. So you can downregulate receptors, you can do receptor-mediated endocytosis. So it's some sort of receptor-mediated issue. But the first three are probably the three most common. We just actively pump it away through active transport. We actively degrade it through some sort of enzyme, or we just let it diffuse away on its own, so it's no longer active. So those are kind of the major ways. And then there's, always some potential for some target cell regulation for the receptors through upregulation, downregulation, or something like that. For the group problems, right? Like always, they're going to be important, okay? If you looked at them, you would see that not quite, but two of the questions from group problems are sort of the questions that you see on that take-home exam that's due Wednesday night, right? And again, I gave that extra assignment I sent out this morning just so that you had an opportunity to figure out how to put that together and upload it, so you're not hopefully nervous about uploading the other one on time and things like that. So the way to look at the exam is, study like you would before. So the big three things are the lecture notes, right? The lectures themselves, which from class, or the ones I posted on Canvas, links to YouTube. Remember the YouTube one, right? You can make it go faster, you can make it go slower, so you can change the speed. And I know somebody says I watch them at 1.5 and it goes real, but maybe the first time you might not want to do that, but it's kind of a nice way to do it. You can just listen to the audio as well, but I am interacting with the PowerPoints and things. Again, if you need any clarification on stuff, let me know, here or later as you study. Like I said, most of us are homebound, so, I'll be checking in every once in a while, most of the day and tomorrow. So you can do that. But so the big two are that. Much more for the notes than the slides. The lecture, so those are posted. and then the group problems, which have always been important, right? As we saw with the first exam, group problems were important. So I think that focusing on the group problems would be beneficial in terms of being able to answer the question. So yeah, and remember, we're running out of time. Half of you to 2/3 of you are going to have to, take that exam, in 18 hours. But writing out the group problems probably would help, or at least portion of them, and just make sure you knew them really, really well. That would certainly help. And answering the multiple choice questions as well. One other thing, since while I'm thinking about it, and I was going to post this, I will, but I keep on forgetting, to be honest. I've got a ton of things I'm juggling right now. Probably you all are anyway. We had a matching section that was going to be on the exam, and I actually have all the exams in my trunk for pen and papers. I got 80 lab quiz 2s and 80 lecture exam 2s in my trunk in my car. But I had a section on matching, and so I'll give you the answers, and I'll post these later, but I'll give you the answers. 1, 2, 3, 4, or more than 5, I believe, are the answers. So I just made those into multiple choice questions and just had the same answer every time. So the last number of multiple choice questions, you'll just see, the same answers every time, because that's like the matching. And it's easier to do it that way through multiple choice than it is. So when you see, God, these look like the same answers, they are. So part of the thing you should do while you're studying the next, rest of today, and, if you're in the night class, probably some of tomorrow, is figure out what those would be, right? So, and I probably didn't ask stuff this easy, but maybe I did. How many branches to the ANS are there, right? Let's see, there's a sympathetic and parasympathetic, that would be 2, right? 2, and things like that. So those are the type of questions that you'll see in that part of it. So question about making my videos public so I can view them without being in class. As long as you have the link, right, then you should be able to get them, right? So you could email me and I'll just send you the links back. That might be the easiest way to do it. So that would be probably the easiest way to do it. Having them searchable. There were some issues, recommendations from us from some of the IT people I talked to keep them private with just the link, and it saves some issues down the line. So if you're in this, you should be able to probably be email me. My email, for those of you that don't know it, is just jhoffman@dvc.edu, 2 F's, 2 N's. And you can ask me for the links to that stuff, and I can do it. That's probably the best way to go about that. And plus all of the, I think most of the instructors have access to that as well, for different things. So that's probably the easiest way to go. But if you got big issues, you can email me and we can discuss it. All right, anything else?
I have 2 questions, actually.
Oh, I like that.
Okay, so the first one is the quickest to answer, I think. For just to clarify about the action potential, so are we going to count it as starting at rest at -70 all the way up and back down, hyper, and then back up to -70 again? Like that whole thing is one action potential?
So it's a good question. Up until from -70 to -55, even though it's a graded potential, if it causes an action potential, it's considered part of the action potential. If it doesn't, it's still considered a graded potential. Does that make sense? So that first part from minus 70 to minus 55 that deflects up, it's considered part of the action potential once you get an action potential, which
Right.
is kind of
So
confusing.
Okay, I understand. So in order for the action potential to happen, the grade potential has to happen. So that becomes part of it for when we're making our graph for the
Yes.
test. Okay, thank
Yes.
you. Second question is, I'm wanting to know how much detail, the level of detail we need to know about the adrenergic
So you should be
receptors.
able, yeah, that one's you should know pretty good details on that.
Okay.
So
Okay.
let's go over it.
Okay.
Right. And again, I was confident that many of you would show up, but I wasn't sure. So um, I scheduled it for this thing for only a half hour, but I'm willing to keep on plugging away if you got questions. So oh, that's the wrong one. So let's look at neurotransmitters, right? Actually, let's go up one more. No, this one's fine. So the question was asked about neurotransmitters, and we'll go in more detail in a minute. But in the peripheral nervous system, there's not only two neurotransmitters, there's more. As an example, there are certain cells that have ATP released as a neurotransmitter, things like that. But there are two types of neurotransmitters in the peripheral nervous system we're going to worry about. And those are acetylcholine, and so you see acetylcholine down here, and norepinephrine. This is where it gets a little bit confusing for the acetylcholine one. If it's a receptor that binds to acetylcholine, which we just call ACH for short, that receptor is called a cholinergic receptor. You see choline at the bottom and at the back, and cholinergic. So it's a cholinergic receptor. So all cholinergic receptors, by definition, will respond to acetylcholine. Okay. Now we also have two subtypes of cholinergic receptors. The first is called nicotinic, and the second is called muscarinic. We'll talk about those differences in detail in a second. On the other side of the coin, we have norepinephrine. And norepinephrine, what will happen is for norepinephrine, we will see that we have what are called adrenergic receptors. It's a little harder for students to get, but norepinephrine has a close relative called epinephrine, and epinephrine's other name is adrenaline, so that's where it comes from. So it's adrenergic receptors. So any receptor that binds norepinephrine is by definition called a adrenergic receptor. So when we look at these things, right, we have an alpha 1, a alpha 2, a beta 1, and a beta 2. So we'll talk about those subclasses of adrenergic receptors. So let's look at first the acetylcholine receptors. All right. So before we do that, so we'll look at that. I'm just trying to get everything set up, and I'll explain these. So here's the acetylcholine receptor. So acetylcholine is what we call ubiquitous. It's a nice scientific word. It means they're everywhere, right? I did not have that as a fill-in, but for those of you in my class, you would have said, right? That would be a good fill-in, right? I did not ask it in a multiple choice, although I sort of considered it and tossed it for other things. So the cholinergic receptors have two types. As I mentioned before, the nicotinic receptors. And what happened was when scientists first discovered these, they said, okay, well, it responds to acetylcholine. And being scientists, they want to say, okay, what else does it respond to? And so they put a whole bunch of chemicals on it, and the first chemical that actually responded to it was nicotine. So to tie it back with what we talked about in the homeostasis lecture, right? It's what we call an agonist. So nicotine is an agonist for cholinergic receptors, but only nicotinic receptors. So there are two places we see nicotine receptors, okay? One is in skeletal muscle, right? So they're the ones that we look at that release neurotransmitter to a receptor. That receptor is nicotine. We also see it in the autonomic ganglia. So we didn't go over it in detail, but the sympathetic and parasympathetic system largely are made up of two fiber systems, and that second fiber is cell body is in a ganglia. It's a collection of cell bodies outside the central nervous system, and the neurotransmitter release there is acetylcholine nicotinic receptors. Muscarinic receptors, right, respond to acetylcholine and muscarine, and they're found at the end organs of the parasympathetic division. So like where the heart is or the intestines, right? So that's the detail you have to know about the acetylcholine receptors, that they're cholinergic. There's two types, nicotinic. Nicotinic are found on skeletal muscles and in autonomic ganglia, and the muscarinic receptors are found in end organs of the parasympathetic division. Muscarinic receptors are called muscarinic receptors because they responded to muscarine. They don't respond to nicotine. So muscarine is an agonist, which does that as well. So that's acetylcholine receptors, and that's sort of what you should know for that. The next set are a little more complicated in that, yeah, you do have to know stuff. So hold on. So here's and this is again slide 79 on that new PowerPoint I posted. This shows you all the different receptors, but I'm going to go to the next slide, which shows it as well, but also now talks about the receptors. So it's the same picture, it's just a little smaller there. So we'll look at that in a second. So here we see that we have, again, these are called the adrenergic receptors. And again, because they respond to norepinephrine. Norepinephrine is a close relative of epinephrine, which again, interchangeably with adrenaline. So that's where adrenergic comes from. It's hard to say. And it's actually spelled wrong here. So it should be adrenergic, like adrenaline. So when we look at the adrenergic receptors, we're going to talk about the one that's different. This one's kind of weird, and so it's different than the other receptors. So it's located right here. It's the alpha-2. And the alpha-2, if you look at it, is located on what we call the presynaptic membrane. It's located in the same place that releases neurotransmitter. It's very strange. And it's not what we normally think of as a neurotransmitter. Receptor, right? Most of the receptors, right, are going to be on the other side. So I'm going to make those a different color, right? So here's my receptors here for the other ones, right? And there's one over here as well. So this one in pink is very different than the other ones, and it behaves differently. And what happens is, because they're receptors, they are proteins, and protein receptors are subjected to those major things we've talked about before multiple times, right? Specificity, competition, saturation. So on when we look at these things in terms of those factors, if we stimulated the neuron successively a number of times, then we would release a significant amount of acetylcholine. Once these receptors got saturated, it wouldn't do any good to release acetylcholine because it would just diffuse away or get broken up or whatever. It'd be a waste because it has no effect because of saturation. So what happens is these alpha-2 receptors located on the presynaptic membrane, what they do is they have a low affinity for norepinephrine. So they don't respond until norepinephrine becomes a high concentration. When they do, then they bind to this receptor. And what this receptor does is it goes back and inhibits the production and release of norepinephrine because we already have enough in the synapse, so it prevents the excessive release of doing that. So it's another good example. We had that question earlier about feedback, right? Feedback inhibition, that's a good example. And so what it does is helps us store the norepinephrine because if we keep on releasing it, it's either going to diffuse away or we're going to break it down and we're going to have to make more. So it's the one that's kind of odd. The other ones are your normal receptors, right, that are found normally there. And there could be more than one in a specific location, although typically there's not more than two. So the first one we're going to talk about is called the alpha-1 receptor. So that could be any of these right here at the postsynaptic membrane. So anything on this side, right, would be an alpha-2. And the only really place I want you to kind of understand where they are is they're on systemic blood vessels, and their job is to cause vasoconstriction. So if we stimulate a blood vessel, and alpha-2 is the most common receptor around for the sympathetic division. If we stimulate an alpha-2, an alpha-1, then it's going to cause constriction of the blood vessels. And so we'll see eventually it's going to help increase blood pressure, which will be important in most cases. So that's an alpha-1. That's where you have to know where it's at. The beta-1s are very different. They're a different class of receptors. You probably might have heard about them before. They just call them like beta blockers, right? Is an antagonistic for that. For the beta-1s, their receptors are located on the heart. So they're only found on the heart. There's also, actually, that's a lie. They've fairly recently found them in kidneys, which makes sense. And stimulation of them, right, causes what we call a positive chronotropic effect, which really is a nice way to say increase, if I could write it, I'm getting better, but still not good, increase heart rate, right? So that's the chronotropic. Chrono refers to time. Inotropy, right? That's going to be an increase in, I'm just going to put STR, strength. So for the inotropic effect, we get an increased strength. So the heart actually pushes harder, pushes more blood out. That's a positive. And dromo, anytime we see the word dromo, refers to conduction velocity. So we get an increase in conduction as well. So all of those things are going to help us increase the activity of the heart for like when we're exercising, okay? So these neurotransmitters, right, are always going to be released that we're talking about now at the end organ. So at the heart or at the sweat gland or in the kidney, whatever it is, those are going to be where those things are released. So positive ino, chrono, and dromotropic effects. So that's cardiac muscle primarily. The third type of receptor we're going to find on the postsynaptic membrane are beta-2 receptors. And beta-2 receptors, the two places I want you to know where they are, really the two places that are important, are on the coronary blood vessels. There's really not very many on systemic blood vessels, and to be honest, they don't have much of an effect, in your bronchioles, okay? So those are the two places we see there. And what they do is they relax smooth muscle. So this is a great example where, again, that it's the receptor that gives the effect. In the alpha-1, we saw that it causes vasoconstriction, right? So the alpha-1 constricts blood vessels. It causes smooth muscle contraction, while the beta-2s, right? The beta-2s cause vasodilation. So they relax smooth muscle. And that's important in the heart to get more blood through the heart so that you can get more oxygen to the heart so it can do stuff. And it also has bronchodilation. So vaso refers to vessel, blood vessels. Broncho or bronchial refers to your tubes to your lungs, right? And when you exercise, you want more blood to go to certain areas, So that's why we have beta-2 receptors there. And we want more air going in and out of your bronchioles, so there's less resistance. So you want your bronchioles to widen so we can easily get air in more. So those are the big receptors that we see and we have to worry about in the peripheral nervous system for the sympathetic system. So we've got the alpha-2, which is the weird one. That's the presynaptic membrane one that inhibits excessive norepinephrine release by feedback inhibition. And then the other three are on the postsynaptic membrane, the alpha-1, which causes vasoconstriction in blood vessels, systemic blood vessels. The beta-1, which is located for us primarily on the heart, but also in the kidneys. In the heart, it causes an increase in the heart rate, strength contraction, and conduction velocity. So the conduction. So the heart rate is chronotropic, the strength is inotropic, and the conduction velocity is dromotropic. Just as an aside, we talked about muscarinic receptors in the heart, and that's going to have the exact opposite effect. So it's going to have a negative chronotropic or negative inotropic or negative dromotropic. So those cholinergic receptors, the subtype, the muscarinic, are going to slow things down in the heart. And you might have seen that in lab when we put acetylcholine on the frog heart. It looked like it went slower and weakened, and epinephrine or adrenaline increased it a little bit. And so that's the reason why is we're affecting one receptor versus the other. We didn't see big changes because those are located primarily on the interior of the heart, and we're sticking things on the exterior, so we don't get as big a change. And then the beta-2, those are in the coronary blood vessels and the bronchioles, and they cause dilation of those, which increases flow. All right, so that makes sense?
Yeah, thank you. Really quickly, could you explain conduction velocity? Does that just mean how fast the electricity in your heart is
Perfect.
going?
Yep.
Okay.
Perfect. So in order for your heart, and we'll look at it, we'll talk about it, at least we won't look at it in lab unless we're back at school by then. But one of the experiments we're going to do, we're going to look at the ECG and see how the ECG changes when you at rest and exercise. And when your heart's beating faster, obviously it would help if the actual electricity moved through it faster. So exactly what you said, the conduction velocity is how fast does it move from one end to the other to get the heart to contract. And so it makes sense, it moves faster when you're exercising. Perfect.
Thank you.
All right, anything else? Anybody else? Anything? Going once, going twice, anything? All right, so I got nothing else unless somebody's got a question. Okay, so email me if you got questions tonight. We actually literally could hook up a Zoom thing if you needed to as well. Not very difficult. So thanks, I'll come and hang in there, and I'm gonna shut you off then. So email if you got issues. And I'll see you online tomorrow morning at 8 or at 5:30. Wait.
Thank you.
You're welcome. See ya.
Yeah, thank you.
Bye, everyone.